A phylogenetic tree is a visual representation of the relatedness of species by descent from a common ancestor. The branch tips represent existing species. Branches connect the common ancestor, and the nodes depict where one species diverged into two or more species. Because the fossil record is incomplete and we do not have direct knowledge of evolutionary history, a phylogenetic tree must be inferred indirectly from data. For example, homologous structures, those that are derived from the same body part in a common ancestor, can be examined. The forelimbs of many animals have similar bone patterns. The feathers of modern birds are homologous with feathers found in fossils. In contrast, wings of birds and dragonflies are analogous. They are derived from different ancestral structures. The cornerstone of tree building is to identify shared derived traits accurately. Species that share derived characters belong to a clade. A derived character shared by clade members is called a synapomorphy of that clade. One of the most basic ways to distinguish between ancestral and derived traits is by outgroup analysis. In this approach, traits of the group being studied, the in-group, are compared to traits of a very close relative that clearly branched off earlier, the outgroup. A cladogram is a phylogenetic tree inferred by clustering synapomorphies. In general, cladograms only indicate the order of evolutionary branching events, not the timing of these events. How do we decide whether a tree is more or less likely to be correct? Shared derived characters may have evolved independently as the result of convergence, or may be lost within a group as the result of reversal. For example, adult frogs do not have tails, a trait that unites gorillas and humans. Grouping frogs with salamanders is favored because it requires loss of only one trait, the tail. A phylogeny in which frogs are most closely related to gorillas and humans requires loss of tail, loss of hair, and gain of amniotic membranes in frogs. In the 1970s, researchers began to use molecular characteristics such as comparisons of DNA, ribosomal RNA, and protein subunit sequences to construct phylogenetic trees. These molecular methods have revolutionized phylogenetics. For example, living organisms are now viewed as being divided into three domains: bacteria, archaea, and eukarya. Molecular methods have resulted in a dramatic rearrangement of the protists. Although the fungi are recognized as a kingdom, the algae are now viewed as being scattered among various groups of protozoa. Red algae appear to represent a distinct group, while the green algae are closely related to plants, and the brown algae appear to be related to a specific group of water molds.
Tell me pretty lies, look me in the face. Tell me that you love me, even if it's fake, 'cause I don't fucking care.